A big hello to all my chutku people out there. I hope you're doing wonderful and your preparation is going on really good. So here I am with paper 2 solutions, my dear children. Paper 2 as in what paper, ma'am? So at Deeksha, we are providing you 5 free mock tests. If you do not know about that, let me tell you, listen to me out carefully. So 5 free full-length mock tests is there, which includes questions from all the chapters which are there in physics, chemistry as well as biology. In biology, obviously, there is a segregation into botany and zoology as well. Yeah, so make sure you are practicing these mock tests. If you have not done it yet, please go get yourself registered. As I said, link is there in the description section. Yeah, now, so this solution is a full-length paper 2 solution, okay, zoology part solution. Botany I have already done it. So if you have registered and if you tried and you couldn't do all the questions by yourself, then here I am to help you with that. But I definitely recommend you to try it first with a limited amount of time, like put a 45 minutes timer and do the questions, okay. Put 45 minutes timer and solve one particular part of it, because for each question you just get what, 1 minute, my dear children, not beyond that. So make sure you are doing that, yeah. And if at all you're not able to do, I am here to help you out. Listen to me out carefully. And children, if you're liking the video, do not forget to click on the like button, and also do let me know if you have any doubts. I'm here to solve all of it. So first question, the cross section of the body of an invertebrate is given below, okay. Identify the animal which has this body plan. What is this body plan, my dear children? So basically I can see that there is a layer outside and this layer is known as ectoderm. Okay, then there is an inner layer. What do you call this? Endoderm. Endo as in inside, yeah. And in between them, there is a sandwich layer that is known as mesoderm. Now I can see 3 of them clearly. There is no cavity. There is no cavity up here, my dear children. So usually what happens, whenever there is a cavity like this, okay, this is a cavity like this. In what? In the mesoderm layer, you call these organisms as coelomates, as in they have a body cavity. There is a cavity and this cavity is lined on the either side. If you see, on both the sides it is lined by the mesoderm. So you call them as coelomates. But over here, apparently I cannot see any cavity. The mesoderm is just intact like that, and these organisms are known as what? They do not have the cavity. So A, coelomates. A as in coelom, A is as in absent. Coelom is basically absent. That is what you get to understand from this. Now apart from that, there is also one more group of organism who have something like this. So this is the ectoderm, this is the endoderm, and in the middle they have pouches like this. They are known as pseudocoelomates. Pseudocoelomates, as in there are cavity and this cavity is partially lined by the mesoderm, but not completely. So I call them pseudo, as in false coelom. Even though there is a cavity, it's not completely just like a true coelom that we have in the case of coelomates. Okay, now what are acoelomates, what are coelomates, what are pseudocoelomates? You got to understand that, but you should be knowing their examples also. So the first 4 phylum, Porphyrians, okay, and then Cnidarians, Ctenophora, and then Platyhelminthes. All of them are acoelomates. So over here, these organisms, from this organism, only the ones that belongs to this particular phylum that I spoke about, they are acoelomates, none of the others. So cockroach, my dear children, they are arthropods. Did I talk about arthropod when I was talking about acoelomates? No. So arthropods are coelomates, okay. Arthropods are coelomates. What about roundworm? Roundworms are pseudocoelomates. Roundworm come under Aschelminthes. So after Porphyrians, Cnidarians, Ctenophora and Platyhelminthes, they all are acoelomates, right? Then comes one group of organisms, that is Aschelminthes. They are the only one to have pseudocoelom. Yeah, after Aschelminthes, you have Annelida, Arthropoda, Echinodermata, Mollusca, this and that, everything. All of them are coelomates. Okay, so 4 of them acoelomates, 1 of them pseudocoelom, and rest of them, coelomates, proper coelomates. Now, roundworm, as I said, it is Aschelminthes, so it is pseudocoelomate. Taenia is what? Taenia solium is basically a Platyhelminthes, and Platyhelminthes, my dear children, they come under acoelomates. So this is the answer. What about earthworm? Earthworm is Annelida. Earthworm are coelomates, not acoelomates. Okay. So please remember that 4 people, Porphyrians, P square and C square, that is the thing. Porphyrians and Platyhelminthes and Cnidarians and Ctenophorans, they are acoelomates. Over here, Aschelminthes is the only one, and rest all of them are coelomates. Works? So this becomes your answer. Okay, fine. Now select the incorrect statement. According to 2011 census report, the population growth rate was less than 2%. Okay, is that correct or incorrect? It is correct. You'll get to understand it is correct when you read the next statement. In all assisted reproductive technologies, fertilization occurs outside the female's body. Do you think in all the assisted reproductive art, okay, do you think all of them, in them, one thing is common, that is fertilization is occurring outside the body? No. What about gift? Gamete intrafallopian transfer, what are you doing? You're basically transferring the gamete from a female who can produce it into a female who cannot produce it. But fertilization is happening where? Inside the female body, not outside. So can I say in all assisted reproductive technology, fertilization occurs outside the female body? That is a wrong statement. Not all of them necessarily, okay? Now, creating awareness about sex-related aspects in an effective method is an effective method to improve reproductive health of the people? Obviously, yes. Now, continued intense lactation could help as a natural method of contraception up to maximum 6 months from postulation. As in, for example, I'm a mother. We'll imagine some other person. Okay, there's an X person and this X person just gave birth to a baby. Yeah, and now I can say that this particular person would be naturally, preventing conception because she is lactating. Because she is what? Lactating, my dear children. Why? Because in lactating mother, a hormone is increased way too much, and that is prolactin. Was it? What was it? Che, what is it? It is prolactin. And this prolactin, my dear children, goes and acts on certain other, does not allow the secretion of gonadotropins. Does not allow the secretion of gonadotropins. What are those? FSH and LH. Which is why what happened? A normal ovulation cycle would not be happening. Which is why if ovulation is not happening, if egg is not released, do you think the sperm will come and fuse with anything? No, my dear children. So lactating mothers up to 6 months from parturition, they have said that their body itself acts as a natural method of contraception because basically they are lactating. But the chances of failure is really, really high. Okay, the chances of failure is really, as in people do get conceived. Okay, people do get conceived even while they are lactating. So that is also there. So it's a method with high rate of failure. Okay, you should remember that. So even if you do not know this particular statement, my dear children, if you go ahead with the next statement, which is pretty much clear, you get to know your answer as well. Okay, so always read all the statements carefully, you'll get your answer. Now, which of the following is or are not the characteristics of class Osteichthyes? Osteichthyes means you're talking about bony fishes, not cartilaginous fishes. The fishes in which their endoskeleton is made up of cartilage, you call them chondrichthyes. Okay, now the fishes whose endoskeleton is made up of bones, you call them Osteichthyes. So body is streamlined and mouth is terminal. Will that be correct? Yes, that is correct. Gills are covered by operculum. Is it covered by the operculum? Yes, my dear children, that is also correct. Skin covered with cycloid and placoid scales? No, my dear children, wrong. Cycloid and ctenoid scales. Ctenoid as in like this, it would have been better. Not placoid scale. Placoid scale is something that you find in what? You find in chondrichthyes. Remember that. So this statement is a wrong statement. Okay. Now we have to find the wrong one, right? Yeah. Now many of them are viviparous. No, most of them are oviparous. Most of them are what, my dear children? Oviparous. That means they lay eggs. Mostly. Okay, mostly they are gonna lay eggs, my dear children. So what can I say? What can I say? 3. So it's not like I didn't tell you that basically they are just oviparous. I told you most of them are oviparous. That means they are viviparous also. But mostly if you say, it will be oviparous. So here the opposite statement is written, many of them are viviparous. Wrong. Okay, many of them are actually oviparous. Little of them are viviparous as well. So can I say 3 and 4 are the incorrect statement? Where do I find it? I find it over here, so which is why this becomes the answer. Works? Next question. Consider the following statement. External ears are absent in frog. Only tympanum with internal ears aids in hearing. Yes, they do not have you seen any frog like this with ears like us? No, my dear children. They just have the tympanum, that is the eardrum, and that is the ear. That is the ear. That represents the ear in the case of frog. So that is totally correct. Now the eyes of the frog possess a single unit, hence are simple. Even though when we go ahead, we find that it might have compound eyes and things like that. But we say, since they are using only single unit, this is a line from your NCRT, so we have to believe them. Yeah. So the eyes of frog possesses a single unit, hence they are simple. Always remember, frogs are simple. Frogs are simple. Okay. I know if you just go in detail, you might get other answers. But according to NCRT, this becomes your answer. So we have to say both 1 and 2 are true. Okay, works? Okay. Next. For nucleic acids, the building block is the nucleotide, which has 3 chemically distinct components. In the DNA, which are the following component of nucleotide is present? So in a nucleotide, which is the building block, what all do you find? You find a pentose sugar. This would be a deoxyribose sugar. Deoxyribose sugar as in 1, 2, 3, 4 and 5 carbons are there in this pentose sugar. Yeah, this is a ribose sugar. And why do I call it deoxy? Because usually in the case of RNA, they have an OH group over here. But in the case of DNA, my dear children, there is no O. That is why it is known as deoxy. So they just have a H group over here, right? On the first carbon, they have a nitrogen base attached to them. On the fifth carbon, they have a phosphate group attached. Together, this whole structure is a nucleotide. Now, you are asked in DNA, which of the following component of the nucleotide is present? What is this? This is uracil, my dear children. Uracil is not present in DNA. Uracil is what? A nitrogen base, and it is present in what? In RNA, not DNA. Talking about this, if you see, there is an OH group at the second position. Can it be a part of DNA? Wrong. Now, what about here? Again, an OH group on the second carbon. Can this be a part of DNA? Wrong. What do you have over here? This is adenine, and this becomes your answer. Okay, this adenine is what? A nitrogenous base. So, yeah, I know it is kind of confusing, but yeah, you have to remember it. So, this is uracil and this is adenine that you have, my dear children. Over here, there's an OH group, which is why you cannot consider it to be DNA. Similarly, over here also, there's an OH group, cannot consider it to be a DNA. Now, the same position, my dear children, if you have CH3, a methyl group added to uracil, my dear children, it becomes thymine. Thymine is what? A modified version of uracil, and thymine is the one that is found in DNA. Uracil is the one that is found in RNA. I hope you remember that, yeah? So, what are the purines and pyrimidines that we have in the DNA? Can anyone who is watching this video, who have reached until now, can tell me what are the purines and pyrimidines that are found in the DNA? Okay, tell me that. Let me see who all knows the basic. So, what is the answer, my dear children? Your answer becomes this one. Let's go ahead with the next question. So, in Drosophila melanogaster, the genes white and yellow shows dash recombination. White and yellow, my dear children, suppose the gene is over here, the gene is over here. Why do I say that the genes are really close? Because the amount of recombination that they show is just 1.3%. That means they have to be close to each other. That means they are recombining less, they are showing very less recombination. If suppose this part of the DNA is going, if this part of the chromosome is going, it's going together, which is why both these genes are together always. Mostly they are together. That means less amount of new combinations will be formed, which is why 1.3% recombination is only being shown. Now, what about white and miniature wing? How much percentage of recombination do they show? We have studied from NCERT, it is 37.2%. That is quite high, too much high amount of recombination. That means the genes are located far away from each other. So, white one will be here, but the miniature wing gene would be here, which is why they are showing what? More amount of recombination. This part of the chromosome can move there, can form new combination, which is why we say there is high amount of recombination. But are you asked about recombination percentage over here? No. You are asked the genes white and miniature wing show dash amount of linkage. And how do you find the percentage of linkage? You're gonna minus this from 100. When you minus the rate of recombination from 100, you get your answer, and that would be approximately 62.8%. Okay, so over here you are asked about recombination, that is 1.3%. Over here you are asked about the percentage of linkage, it would be 62.8%. Okay, so this becomes your answer. I would have marked this because I also thought that they are asking about recombination over here, but that's not the case. They are asking about linkage, which is why this becomes your answer, not this one. Okay. Now, the vital capacity of lungs is what? What do you mean by vital capacity of lungs? The maximum amount of air a person can breathe out after the maximum inspiration. Okay, so I take maximum breath in. Yeah. What is this represented by? This is represented by inspiratory reserve volume. Yeah. IRV. Now, maximum amount of air I can breathe out, that is expiratory reserve volume, my dear children. And every time when we are saying about IRV and ERV, we are also talking about TV, that is tidal volume. Tidal volume is what? The amount of air that we breathe out in a normal breath. Breathe in and breathe out. So, all of it together becomes the vital capacity of the lungs. So, I will say IRV plus ERV plus TV will become your answer. Okay. Next, identify the correct statement for human heart. Volume of both atrium is greater than the volume of both ventricles. So, this is the atrium that we have, and this is the ventricles that we have. Do you think the volume of both atrium is greater than the volume of both ventricles? Wrong. Volume of both ventricle is greater than the volume of both atrium? Correct. What do you have to find out? You have to find the correct statement. So I found one, but let's see the others. Interventricular septum separates right and left atrium. Interventricular, that means it is between the ventricles, not between the atria. So how can they separate atrium, left and right atrium? Wrong. Atrioventricular septum, that means septum between atrium and ventricles, okay, separates right and left atrium. Array, again wrong. So what is the only correct statement that we have? It's B. Now, next question. The maximum amount of electrolytes are reabsorbed in. The maximum amount of electrolytes, my dear children, reabsorption takes place in none other than proximal convoluted tubule. Ma'am, what about ascending limb of loop of Henle? There also it happens, but you're asked about the maximum amount of electrolytes that are reabsorbed. It happens in proximal convoluted tubule. So from here, from the proximal convoluted tubule, right, certain electrolytes would be passing from the tubule to the blood vessels. So maximum amount of reabsorption, okay, the blood vessels is taking these ions back. Okay, this happens in proximal convoluted tubule and it is an active transport process. That means you need to spend ATP to transport things, but it happens. Okay, so that is your answer. Second best would be ascending, but over here you are asked about the maximum amount, so this will be the only answer that you have. Next, ultrafiltration is due to pressure in the glomerulus because the diameter of efferent arteriole is greater than. So my dear children, suppose this is the Bowman's capsule that we are having. Yeah, over here there is your efferent, starting with A, efferent arteriole coming in, and they make a bunch of capillaries up over here and they move out. But this time, this particular thing that you see, that is efferent, E for exit, okay, this efferent arteriole has a narrower blood vessel. It is narrower. Why do you think it's narrower? Why do you think the diameter is less over here, such that here it is more? So blood will come like this, but it cannot easily pass through that, which is why it will stay here for some time, and that's the purpose. Over here it has to stay and pass down the filter. This is known as glomerular filtration. If it would have been bigger, my dear children, same amount of blood will go. Will it stay over here? Do you think this will happen? Glomerular filtration can happen? No. And this is known as ultrafiltration also. So ultrafiltration is due to the pressure in the glomerulus because the diameter of efferent arteriole is greater than what? Efferent arteriole. This is artery, this is also arteriole. No, both of them are arterioles. Please remember that. So efferent arteriole, E for exit. The exit one is always, okay, you might have seen in entrance. Entrance is always very good, very grand. Exit will be small place out there, right? So A, you can remember like that. A as in A, E as in. Read the following statement and choose the correct option. Cerebellum has a very convoluted surface in order to provide space for more neurons. Do you think? Cerebellum is a part of what? Your hindbrain, my dear children. Yeah, over here you have your cerebellum. If you see the structure of the brain, my dear children, you have the brainstem just behind back, you have the cerebellum portion, right? So this is highly convoluted surface because this has a very highly convoluted surface in order to provide more space for neurons. Yeah, answer is correct. The medulla is connected to the spinal cord. So over here you have medulla oblongata and also you have pons. So medulla, my dear children, do you think it's connected to the spinal cord? Yes. That is also correct. Okay, what do you have to find out? You have to find the correct option. Now, when a neuron is not conducting any impulse, that is the axoplasm inside the axon contains high concentration of K+. So when neuron is not doing any work, it's saying that there is high concentration of K+, and there are negatively charged proteins. There would be negatively charged proteins out there and low concentration of Na+. There will be less concentration of Na+, which is why I'll say, even though K+ is there, but since there are so many negatively charged proteins, my dear children, I would be saying that there is what? Negative environment over here, and this negative environment is minus 70 millimoles, which is actually the resting membrane potential, and that is absolutely correct. It's because there is low sodium. Okay, there is potassium, I do agree, but there are more negatively charged proteins and there is less sodium, which is why it is totally negative. Resting membrane potential is totally negative. It is minus 70 millivolts. Fine. So can I say 1, 2, 3 statements? All of them are correct. I can say that, right? So all are correct. Your A becomes the answer. Now, next. The posterior pituitary gland is not a true endocrine gland because, why do you think? There are two lobes of pituitary gland. Okay, there is anterior and the other one is posterior. Okay, so why do you think that posterior one is not considered to be a true endocrine gland? The thing is anterior one will be releasing certain hormones and things like that, but posterior ones actually stores the hormones. Does what, my dear children? Stores the hormones made by the hypothalamus. So hypothalamus makes some hormone, sends it over here, stores, this guy stores it over here and also releases it. Stores it and releases it. Okay, that is the function, which is why it is not known as a true endocrine gland. So it is provided with duct. It only stores and releases hormones. It is under regulation of hypothalamus. It secretes enzyme. The thing is, it only stores and releases hormones, which is why it is not considered to be a true endocrine gland. This one actually makes their own hormones. They have their own hormones. It is being released when hypothalamus say that you have to release it. The command comes from here, but they are the one that make it. So here it's not like that. They are storing and they are releasing someone else's hormone. So yeah, I hope you got it. Okay, and examples of that is ADH. Okay, antidiuretic hormone is an example of this, and then oxytocin is an example of this. Cool. Now, the law based on fact that the characters do not show any blending and both the characters are recovered as such in F2 generation, although one character was absent in F1 progeny is what? What law does it suggest? So basically, I had crossed tall plants and dwarf plants. In the F1 generation, all of them were tall. All of them were tall, my dear children. In F2 generation, I got a mix of, let's say, 3 of them were tall, 1 of them were dwarf. What law do that suggest? So basically, so over here, all of them were tall, none of them were dwarf. But in F2 generation, I found that 1 of them was dwarf. Did I find it over here? No, but I found it over here. So what law does this suggest? This is law of segregation. Okay, this suggests law of segregation, according to which, my dear children, when you're talking about the gametes, suppose these are the chromosomes that a person is having, a plant is having, it is going to form 2 types of gametes, and over here, each chromosome is going to go to each gamete. So this is capital T and this will be capital T over here. Okay, similarly, since they have the similar chromosomes, similarly same thing happens over here. Okay, so all of them, when this and this combines together, you get capital T small t, understandable. Now when you are going to cross this plant, capital T small t, again with another capital T small t, what is going to happen? Are they going to go together? Are they going to go together? No. Basically, what is going to happen is that they are going to segregate. They are going to separate from each other. One of them will be going in one gamete, another will be going in another gamete. Similarly, one of them in one gamete, another one in another gamete. Now, this and this fuses, you get capital T, capital T. This and this, you get capital T, small t. This and this, capital T, small t. This and this, small t, small t. Did I see dwarf children over here? Yes. But did I find it in F1 generation? No. So, isn't it because of some law that they are following? Yeah, and that law is law of segregation. They segregate, they are not moving together. They are going to segregate and they are going to move. Now, children usually have confusion with law of independent assortment and this. Usually, we say independent assortment is for dihybrids when you're talking about 2 characters at one. Here you're just talking about height, right? But when you're talking about 2 characters, or in the question it is mentioned about 2 characters, that is basically I got parental varieties, yeah, and also non-parental varieties. I had crossed round and yellow and wrinkled and green, but I got round and green. That was not parental. Okay, so all of this, they suggest law of independent assortment. That is law of independent assortment, where one gene is going to independently assort from another gene. That is the thing. Over here, talking about law of segregation, where one chromosome segregates from another chromosome. Only one chromosome are going to get into each thing. And the genes also, they are going to separate from each other. Only one gene is going to go into one particular gamete. So your answer would be law of segregation. Next. This question would be needing a little amount of brain, which is why I put in red. So 2n is equals to 16 is in a primary spermatocyte. So primary spermatocyte, which is having 2n, my dear children. The 2n means you're talking about they have, this is the chromosome number, okay. Now, which is in metaphase of first meiotic division. What shall be the total number of chromatids in each of the secondary spermatocytes? So, I hope you know that primary spermatocyte undergoes meiotic division and forms what? Secondary spermatocyte. Okay, secondary spermatocyte. And each of the secondary spermatocyte will be n and n because it becomes half. It was diploid, it becomes haploid. So, ma'am, if this was 16, it would have been 8 now. Half of 16 is what? 8. But are you asked about the total number of chromosomes? No. You're asked about something else. What shall be the total number of chromatids, not chromosomes, my dear children? So basically, when I start with meiosis, always remember, if I have 16 chromosomes, these 16 chromosomes, basically all of them, all of the DNA which is there, it has replicated, which is why it looks like this now. So this is one chromatid, this is sister chromatid that they have. So you have one chromosome like this, similarly another chromosome like this. So you would be having 8 of them on this side, 8 of them arranged on this side. 8 of them on this side, 8 of them arranged on this side. Total, if you say, it is 16. Because you do not count this as 2 chromosomes. You say they are 2 chromatids, but if you are talking about chromosome, this is only 1 chromosome. So this is 1 chromosome and this is also 1 chromosome. In total, how many chromosomes will be having? If 8 of them are over here, 8 of them are over there, 8 plus 8, it is 16. It is what? 16. Now what happens in the case of meiosis? After meiosis is done, okay, after meiosis 1 is done, you get 2 cells. You get 2 cells having what? 8 here, 8 here, agreed? Okay, so what is the number of chromosomes? Ma'am, 8 only. Here it's 8, here it's 8. So your answer is correct? No. You're asked about number of chromatids. And how many number of chromatids are there? Each chromosome, my dear children, it has 1 chromatid over here, 1 extra chromatid over here. So 8 of them if are there, all 8 of them will be having 2 chromatids like this. 8 into 2 will become what? 16. 8 into 2 will become what? 16. Always remember that. I'll come back once more. Listen to me carefully. The total number of chromosome is 16. Is 16 over here. Number of chromosome. 8 of them this side, 8 of them that side. 16 chromosomes. Okay. Now, after meiosis 1, they're going to divide into 2 cells. 8 goes here. 8 goes here. Are you asked about number of chromosomes? No, you're asked about number of chromatids. How do we find that, ma'am? Basically, each of them will be having 1 sister chromatid each. So, can I say 8 into 2 is equals to 16? 8 into 2 is equal to 16? Yes. So, can I say if in each of the secondary spermatocyte, it will be 16 number of chromatids? Yes. If you're asked about number of chromosomes, it would have been 8. Number of chromatids is 16. Okay, if you are asked about number of chromatids in the case of primary spermatocyte, what will be your answer? What will be your answer for primary spermatocyte? Listen, 8 plus 8, 16 of them are there and all the 16 of them will be having one thing. So 16 into 2 is basically 32 chromatids. 32 chromatids. So you are not asked about primary, you are asked about secondary, so that is fine. You just have to talk about this one. Okay, I know this question is kind of interesting. I totally get it and you have to remember it. Okay, do not do any mistakes out there. Cool. Now, if you are asked about, okay, suppose this secondary spermatocyte also divided. It also divided, it underwent meiosis 2. This is meiosis 1, right? It underwent meiosis 2. Now, in meiosis 2, it will divide into 2. This also divides into 2. Now, tell me the number of chromatids in each of them. So, 8 of them were like this. During meiosis, they got separated. Yeah. So 16 chromatids that we had, they got separated. You would have 8 here, you would have 8 here. Similarly, 8 here, 8 here. So all of them would be having 8 of them. 8 chromatids or basically chromosomes. There is no chromatid concept over there because it just looks like this. It just looks like this. They do not have an extra pair there. So they're just chromatids. Really, what do you call them? Chromosomes. That would be better. Okay, so 8 of them, 8 of them, 8 of them, 8 of them. Works. Okay, or if you want to say chromatids, then 8. Chromatids, chromosomes, all 8. Now, question number 15. All of the following statements are correct about active immunity except, so you have to look for the incorrect statement. It is slow and takes time to give its full effective response. Yes, my dear children, it's active immunity where what you're doing, you're making your body release antibodies. And whenever our body encounters something foreign, it will take some time for our body to understand, oh, this is foreign, I have to produce this type of antibody and that and this. So lots of processing time is needed there. So it is slow and it takes time to get fully effective. That is perfectly correct. In passive immunity, what you're doing, you're directly giving antibodies. Your body is not actively participating. Actually, the name is total ulta. Okay, But please understand, in active immunity, you're wanting your body to actively participate, but obviously this time it will be really slow. The first time it encounters, it will be slow. Second time, it will be really fast. Okay. Now, memory cells are formed. Are they formed? Yes. Memory cells are definitely formed, and with the help of this memory only, next time when someone comes to attack, we can fight back against it. So this is also correct. Now, antibodies are not formed during primary response. Do you think antibodies are not formed? Memory cells are formed, antibodies are also produced, but the amount is really less because they are slow, no? So the amount will be really less. So I'll say this is wrong statement. This one is the wrong statement. Now, exposure of antigen is a prerequisite for development of active immunity. Do you think exposure of the antigen should be there? There should be exposure to the antigen, then only our body will be, developing active immunity? Yes, obviously. Yeah, and that is why we give vaccines. If naturally we are not getting any diseases and we want immunity against it, we give vaccines. Okay, that is basically the weakened pathogens or disarmed pathogens that we are putting inside our body, a part of it probably, so that our body produces antibodies against it. So we need exposure to the antigen either naturally or probably we do it synthetically with the help of vaccines and things like that. Okay, so basically that is also correct. The only incorrect statement is C. Now, match the following and choose the correct option. Trophoblast, cleavage, inner cell mass and implantation. What do you mean by that? So basically when zygote is formed, zygote is one cell, single celled, unicellular. It divides into 2, again divides into 4, then 4 becomes 8, then 8 becomes 16 and things like that. Okay, so this division is known as cleavage. This division or mitosis is known as what? Cleavage. So cleavage goes with what? Mitotic division of zygote. So B goes with 4. D goes with 4. Do I have an option like that? I have 2 options like that. So basically I can cut these 2 options. Okay. Now trophoblast. So when I say that they are dividing, they have names. The embryonic stages that it forms. Okay, zygote, 1 cell. Now after that 2 cell, 4 cell, all of them are known as embryos, embryonic stages. You have multiple stages. One is morula where there are 8 cells, 8 to 16 cells. And then come blastocyst stage where, my dear children, there will be outer trophoblast layer. This is blastocyst, okay. There is outer trophoblast and there is inner cell mass over here. There is what? Inner cell mass and outer trophoblast. Yeah, and there is a coelomic cavity like thing over here. Not coelomic cavity, but yeah, there is a cavity over there. This is known as blastocyst stage. Now, what happens to the trophoblast? Trophoblast, my dear children, it is the one that would be attaching to the endometrium, the outer layer of blastocyst and the one that attaches to the endometrium. So A goes with 3. A would be going with 3. So A goes with 3. I have just one option and with that I found the answer for this question. So let's see. A goes with 3 and B, cleavage, goes with what? Mitotic division of zygote. Yeah. And now inner cell mass, what happens to that? So this guy went and attached to the endometrium. What happens to this? This is the one that actually forms the embryo. Okay, so inner cell mass is the group of cells that would differentiate as embryo. And implantation, the process where the trophoblast is going and getting implanted into the endometrium, this is known as implantation, embedding of blastocyst in the endometrium, which is why this becomes your answer. Okay. Next. Read the given statements and select the correct option. Statement 1. MTP is considered relatively safe during the first trimester of pregnancy, as in first 3 months of pregnancy. Do you think MTP is considered to be relatively safe during that? Yes. Statement 2. Fetus becomes intimately associated with the maternal tissue after the first trimester. Do you think the fetus which is there, the baby which is forming, it becomes intimately associated with the maternal tissue after the 3 months? Okay, which is why it is said that before 3 months it is safe to do abortion or induced abortion, that is MTP, medical termination of pregnancy. But after that it is not comparatively safer. Why? Because the baby and the maternal tissues are going to be very intimate. They are going to form placenta. So it's not like placenta formation has not yet started. It has started and it will fully form. Okay, it will be fully formed by this particular time, which is why it is said that you should be doing before. So can I say both the statements are correct? Yes. Okay, both 1 and 2 statements would be correct. Next, which of the following is correct for the condition when the plant capital Y, small y, capital R, small r is back crossed with a double recessive parent. Double recessive parent as in recessive, both of them are recessive. So it's like this. Okay, here one is dominant, other is recessive. Either one is dominant, other is recessive, but here both of them are recessive. Now, what do I find? Which of the following is correct? Let me see. So first of all, I have to cross them. How do I cross them? I have to make the gametes. So how do I make the gametes? So my dear children, for your understanding, always remember the chromosomes which are there. Suppose it's like this: capital Y over here and small y over here. Capital R over here, small r over here. So this chromosome can, whenever gametes are formed, from one pair, one chromosome can go. Suppose this chromosome is going and this chromosome is going. So you get capital R, capital Y, capital R. Okay. And suppose this and this is going. You get capital Y, small r. And then suppose this and this is going. So small y, capital R. Okay. And just give me a second, children. Yeah. Where were we? Yeah. So small y and capital R went. Now small y can also go with small r. So these are the gametes that we have. What about this guy? What about this guy? What are the gametes for them? Let's see. Whatever you do, small y and small y is there, and small r is there. So whatever combination it will be there, the only type of gametes that are going to form is small y and small r. So now we have to cross them. How do we cross them? So let's cross them. Okay. So over here itself I am going to cross. Listen, it's very, very, very easy. Okay, don't complicate things. So capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. Over here we just have 4 type of gametes and all of them are same. Small y, small r, small y, small r, small y, small r, small y, small r. This and this fuses, you get capital Y, small y, capital R, small r. This and this fuses, you get capital Y, small y, small r, small r. This and this fuses, you get small y, small y, capital R, small r. This and this fuses, you get small y, small y, small r, small r. Okay. So this is what yellow and round, because one dominant one is there. This is what yellow and green, yellow and what? Wrinkled. This is what? Green and round, and this is what? Green and wrinkled. So I've got 4 type of phenotypes. 4 of them are very, very different. 4 of them are very, very different. Will I find something else over here? No, because all the gametes are same over here. So whatever I do, I am going to get this only. Okay, I am going to get this only. And all of them basically is 1:1:1:1 ratio, my dear children. The phenotypic ratio is going to be same, and similarly, genotypically also they are going to be the same only. 1:1:1:1 is the thing. So your answer will be ratio of phenotype and genotype would be same, that is 1:1:1:1. Okay, so if you know to make the gametes, that is why I showed you like this, it becomes more easy. You would be able to find the answers in an easy manner as well. Next, next. Which of the following is incorrect regarding ZW type of sex determination? Where do we see ZW type of sex determination? In the case of birds. In the case of birds, you see this. Now, it occurs in birds and some reptiles, correct. Females are homogametic and males are heterogametic. What do you mean by homogametic and heterogametic? Homogametic means they are going to produce same type of gametes. So, over here in ZW, actually the females are heterogametic. They are going to produce 2 type of gametes, Z and W. Over here, the males are homogametic, Z and Z. Okay, so this is a wrong statement. Did you have to find the incorrect one? Yeah. So this is the incorrect one. Let's go ahead. 1:1 sex ratio is produced in the offspring. Yes, half of them would be females, half of them will be males. You want to try it out? Let's see. This ZZ, that means it's a male. This ZW, that means it's a female. ZZ again a male, ZW again a female. So 50% chance female, 50% chance male, so 1:1 ratio. Now all of this cannot be true, so this is the only one that is incorrect. Now, next, sickle cell anemia is an example of. So you should be knowing what all examples are. So sickle cell anemia is because of mutation in one particular gene. So can I say it's a Mendelian disorder? So whenever the mutation in gene happen and because of that someone is getting the disease, I will call this Mendelian disorders. And these can be passed on from generation to generation. I will call this genetic disorder as well. Can I call it chromosomal disorder? Do we increase the number of chromosomes? Is it because there's an increase in number of chromosome or decrease in number of chromosome, things like that? No, it is not caused because of that. Just because one particular gene is mutated, which is why it is occurring. Now, inborn error of metabolism? No, it is not. It is an inborn disease, but it is not error of metabolism. It is not a metabolic disease. It is not going to cause any trouble in metabolism. So this is wrong. Okay. Point mutation, yes. Okay, this is an example of point mutation. Why? Because GAG is transformed to GUG. This point mutation happening over here. Point swap. Frame shift mutation, no, my dear children. Sex linked disease, no. Recessive disorder, yes. It's a recessive disorder. When both the parents are going to give one genes each, both of them affected genes, if they are passing, only then that particular person would be having disease. If only one gene is affected, other gene is not affected, that particular person is not going to have any disease. So both the genes have to be affected. So that is what is a recessive disorder, correct. Qualitative disorder, is it a qualitative disorder? Yes, because the quality of beta hemoglobin chain is being deteriorated. Okay, the quality is being changed up, which is why it is a qualitative disorder. Okay, it won't be able to carry enough amount of oxygen up there. Quantitative disorder, wrong. Autosomal disorder, is it because of the genes on the autosomal chromosomes? We have two types of chromosomes, sex chromosome, X and Y, X and X, yeah, and then autosomal chromosomes. All the other chromosomes apart from the sex chromosomes are autosomal chromosomes. So in that it is happening, yeah, remember chromosome number 11th, on chromosome number 11th, my dear children, at the 6th codon position, my dear children, what is happening? This change is happening. So it is an autosomal disorder. So what other things are there? I'll say 1, 2, 1, 2. Okay, 1, 2, 4, sorry, 1, 2, 5, 1, 2, 5, and then 8, and then 9, and then 11. Yes. So this becomes your answer. Okay, this would be your answer for the question. Next question. According to David Tillman's long-term ecosystem experiments, the total biomass in plots with more species show what? David Tillman did the experiment and then he found that whenever a plot had more number of species, my dear children, had more number of species, what could he observe? Average variation from year to year? No variation from year to year, that cannot happen. No variation cannot happen. There will be some amount of variation, but less variation from year to year, yes, because this is more stable. More species means you are adding more stability to the ecosystem, my dear children. So there will be variation, but this variation would be less. It won't be way too much and it won't be null. It won't be zero at all. Okay, there will be variation, but it will be less. High variation from year to year, wrong. So less variation from year to year is what he found according to David Tillman's experiment. Next, analyze the given pedigree chart and select the option that is correct regarding it. So we have to find out what does this pedigree chart say. Yeah, so let's see. Whenever, I already told you, in pedigree chart it's easy to understand whether it is autosomal or dominant or recessive or is a sex-linked disorder or whatever. How do you find it? First of all, if one of the parents are affected, it is dominant disorder. Okay, if it is not skipping generation, then it is dominant. For example over here, a parent had, children have, now their children is also having. It is not skipping any generation, my dear children, which is why it is a dominant disorder, and at least one of the parent would be affected. At least one of the parent would be affected. In the case of recessive, it's like without the parents being affected, you can see that in the children. So it's skipping the generation. But over here, it is not the case. Okay, in every generation, you can see at least or some organism being affected. So you got to understand that it is what? It is a dominant disorder. That is correct. Now, sex-linked or autosomal? The problem is in the genes on the sex chromosomes or on the autosomes? How do you find it? So basically, sex-linked disorders are found, if a person has that, mostly it would be the males who are affected. Mostly it is the males who are affected. But over here, both of them are equally affected, females and males. The box shows males and the circle shows females. So both of them are equally affected. So can I say that it is autosomal disorder? Yes. So it should be autosomal dominant disorder. So what is an example of autosomal dominant disorder given in NCERT? It is myotonic dystrophy. Under the pedigree chart picture itself it is given. So this becomes the answer. Autosomal dominant, one more disease is given, that is cystic fibrosis. Is it actually autosomal dominant? It is autosomal recessive disorder. So this is wrong. Okay, and autosomal recessive trait phenylketonuria, that is not shown over here. Even sex-linked dominant trait myotonic dystrophy. Myotonic dystrophy is not sex-linked, it is autosomal and it is what? Dominant. Now, next, consider the following statements. Natural selection is a process in which heritable variations enabling better survival are enabled to reproduce and leave greater number of progeny. Is it what you mean by natural selection? Yes. Natural selection is basically I am choosing organisms who are better adaptable to one particular area and they are able to produce. That is correct. Now, Darwinian variations were small and directional. According to Darwin, he said that yes, organisms are not looking same, they have some variation. Okay, they have some variation, but this variation that you see are very small and they are directional. Directional as in always this variation is for good. That is what you mean by directional. Okay. Then came Hugo de Vries. He said that the variations that you see, it's not happening like that. It is occurring because of mutation, some changes in the DNA or whatever. Yeah, and these are totally directionless. Hugo de Vries said that. It is totally directionless. Good can happen, bad can happen. It's not like how Darwin thought. Darwin thought it was small and directional. It is always towards positive direction. So this particular thing is correct. This is what he thought, and this is also correct. So can I say both the statements 1 and 2 are true? Yes. Both of them are true, my dear children. Okay, going ahead. Which of these statements about cancer is incorrect? You have to find the incorrect statement. Cellular oncogenes are present in normal cells which have not become cancerous. Yes, my dear children. They are there in normal cells. We have it in our body. Once some mutation happens, something happens, they become active, and that's when it becomes carcinogenic. Otherwise, they are there normally in our body. This is correct. Antibodies against cancer-specific antigens are used for detection of certain cancers. For example, I am a person. Okay, there's an X person. Okay, and this X person has cancer, and we got to know from their body that certain antibodies are released against these cancerous antigens which are there. So we are sure that these antibodies are released only in patients with what's a cancer. So what do we do? For example, there is Y person and I have to check whether that person has cancer or not. I'll take their antigen and collect the antibodies also, see if they are reacting. If they are reacting, that means what? That person also has cancer, which is why the antibodies are reacting to it, right? So it is cancer specific antibodies. So that is also correct. Now MRI uses strong magnetic fields and ionizing radiation to accurately detect pathological and physiological changes in the living tissue. Now this statement is seeming to be so correct because almost 99% part of it is correct apart from just one. Okay, it is not ionizing radiation, it is non-ionizing radiation, my dear children. That is why MRI is considered to be comparatively safer. Okay, in computers, in what do you call, in X-ray machine and things like that, you use ionizing radiation, but over here it is non-ionizing radiation that you are going to use, which is why it is comparatively a safer option. Okay, so this is the wrong statement that we were looking for. Cancer can be treated by using alpha interferon, which activates the immune system and helps in destroying tumor cells. You know what? They are not actually treated with the help of that, but I'll tell you. So alpha interferons are something that are biological response modifiers. It will boost up our immunity. So people who are having cancer, because of the treatment that they have underwent, so many good cells, so many bad cells have died. Okay, so we want them to be very immune, and for that we give alpha interferons. When alpha interferons are given, it activates their immune system, my dear children, and it is going to destroy the tumor cells as well. So this is also correct. This is the only one that is incorrect. 24th. Now, choose the correct statement about the patterns of biodiversity. The diversity of plants and animals is uniform and evenly distributed throughout the world. Yeah, the tropics, you have to find the correct statement. Yeah, the tropics harbor more species than the temperate or polar regions. Is it correct? Yeah. Read first, okay. Speciation is a function of time as the tropical latitudes have been disturbed by frequent glaciations in the past. So it says the tropical latitudes are the one that have been disturbed by glaciation. Okay. Now D, lesser solar energy in the temperate region, as in colder region, contributes to higher productivity and greater diversity in the species. Do you think in temperate regions there is less, there is less solar energy, and because of which there is high productivity? Can that be happening? No, my dear children. Less solar energy, that means less productivity. So this is correct. Lesser solar energy in the temperate regions contribute to lower productivity, not higher productivity. That will be wrong. Now, speciation is a function of time as the tropical latitudes have been disturbed by frequent glaciation. No, tropicals are comparatively safer. They are stable. It's the temperate which undergo what frequent glaciation and things like that. It is not at all stable. So it cannot harbor more amount of organism. Always remember that. So this would be wrong. Now, the tropics harbor more species than the temperate or polar regions. Yes, as I said, because it is what? It is more stable, which is why, my dear children, it will be harboring more species than temperate or polar regions. The diversity of plants and animals is uniform and evenly distributed throughout the world. No, it is not. Some areas there which have more diversity, some others which do not have it. So this is wrong. What is correct? Only B. Next, Assertion says intrauterine devices are very effective contraceptive method. Intrauterine devices as in something that you put inside the uterus and they release something out there and change the whole environment in the uterus, does not allow conception to happen. So that is correct. It is highly effective and it is one of the most widely accepted technique. So that is correct. It is best for spacing children also. For example, one couples had child, okay, and now they want space like 5 years gap. So yeah, they can have. Yeah, and the reason is IUDs don't allow sperms to enter the uterus. Do you think all the intrauterine devices are like that? Do you think they do not allow sperms to enter? That is how they stop conception? Wrong. You have three types of IUDs. Non-medicated IUDs, which are basically put inside the body. They do not produce anything. But since they are foreign to our body will react to it. And while reacting to it, any sperm that comes along, my dear children, later on, okay, it will be killed. Okay, because something foreign is already there and our immune system is really active. That is non-medicated IUD. Example is lipase loop. Second thing, CUT IUDs, that is copper releasing IUD, not CUT IUDs, copper releasing IUDs. For example, CUT, that is copper T. Okay, in that what happens, the copper is being released and this copper is going to mess up with the motility of the sperm. So sperm which entered the female body, it messes up with the motility of that, it slows it down, okay, such that it does not move really fast, such that it does not go and fertilize there, okay, and it also messes up with the fertilizing quality of the sperm. So that is copper releasing IUD. Again, you are making the entry of sperms there, right? Now last thing that you have is hormone releasing IUD, where they are going to produce certain hormones which are already being produced by our body but in excess amount. And when everything happens in excess, what happens? Our body will react negatively to it. So progesterone and estrogen, which were responsible for ovulation to happen, now when they are released, it wouldn't allow ovulation to happen. The endometrium, which was thick because of them, will now become thin because of the same thing. Okay, and the cervix which was there, okay, there you will be getting a mucus plug such that the sperm cannot enter. So that is only IUD where basically this transport of sperm is basically halted, otherwise it is all good. Okay, so I cannot say IUDs don't allow sperm to enter the uterus. That would be a wrong statement to generalize that. So I'll say this is false and this is correct. Okay, so your assertion is true and the reason is false. Okay. Next, the recombination frequency between the genes A and C is 5%. Okay. So I have A and C. 5% recombination is there. That means they show less recombination. That means they have to be close. So you see, first of all, before going ahead with the question, read which is the highest one. So 29. 29 is the highest one. So, and that is between A and D. So my dear children, before starting of the question, what do you have to do? I tell you. Okay, write it. So A and C is 5%, okay. B and C is 15%, and then B and D is 9%, and A and B is 20%, and C and D is 24%, and A and D is 29%. Now look for the biggest one. It is this. That means you have to keep them at the end. So A and D are going to go at the end because they have how much gap? 29% recombination is there. Okay, that means they are very far from each other. They are going to be really far from each other. Now let's go with the second one. C and D. C and D is again 24%. So this was 29. So obviously C will also be somewhere over here. So that's how this becomes 24. And this whole thing becomes what? 29. Cool. Now what? A and B is 20%. So A and D is 29%. A and B is 20% means B would be somewhere over here. Okay, near D. Yeah. Now B and D is 9%. Yeah, they are very close. That is why it is 9%. Cool. B and C, my dear, 15%. Yeah, that's fine. A and C 5%. That means they are close. So can I say that it is A, C, B, D? The arrangement is what? A, C, B, D. So always try to find the farthest one. It will be easier. So biggest one, it will be easier to identify it. So A and D, then go ahead with all this. Okay, write it down and then do that. It will become easy. Did you enjoy this question? I love this question. I like this type of questions. Yes. Now, which is the correct path of secretion and transport of milk in the mammary gland? So, dal fell on mat. I always remember like that. Mat comes first and then comes dal. Okay, mat was already there, dal fell on it. So, first one is mammary lobes. Inside the mammary lobes, you have alveoli. Inside the alveoli, they come together to form tubules. The tubules, they come together to form ducts. The ducts come together to form ampulla. And the ampulla comes together to form what? Lactiferous ducts. Okay, this should be the flow. So, which is the correct path to the secretion or transport of milk in the mammary gland? Alveoli, cavity of alveoli, if it is alveoli, let's see, alveoli, cavity of alveoli, then mammary tubules, then mammary duct, then mammary ampulla, then lactiferous duct. Is it correct? Yes. This itself is correct, but let's see if we have another option that includes lobes also. There is no option that includes lobes, right? So, I will say this is the only correct option that I have. I am not going to read it ahead. Okay, so always remember this dal fell on mat. I don't know how helpful it is because mammary lobes has an L over here, but this L and that L would be confusing for you, which is why mammary lobe. Let's start with mammary because you're talking about mammary gland, so mammary lobes, then alveoli and everything up there. Yeah, cool. Next, in lactational amenorrhea, there is no ovulation or menstruation during the period of intense lactation following parturition due to high level of prolactin, which does what? So, I told you lactational amenorrhea is when basically a lactating mother would not be menstruating, amenorrhea as in no menstruation. Okay, why? Because ovulation is not happening. Yeah, why is it not happening? Because she is highly lactating and in her body the level of prolactin hormone is way too much. Agreed. But what does it actually do that she does not ovulate? The thing is, this particular prolactin which is there, okay, this particular prolactin which is there, it is going to inhibit the release of gonadotrophins. Gonadotrophins, as in you're talking about FSH and LH, and if both of them are not there, my dear children, you won't see the production of estrogen and progesterone as well. So, where do you expect ovulation would be happening? Follicle stimulating hormone basically makes the follicles mature to ovulate, right? If they are not there, how do you think everything else would happen? So, it inhibits the release of gonadotropins, which is why estrogen and progesterone also get not released, and the other things. First thing is this. Okay. Now, next, which is incorrect about color blindness? What is incorrect about color blindness? This is due to defect in either red or green cone of eye, resulting in failure to discriminate between red and green color. Correct. A daughter will not normally be color blind unless her mother is carrier and her father is color blind. So, daughter would not be color blind, yes, until her mother is a carrier and the father is what? Color blind. Yeah, because one from the father, you are going to get one from the mother. So, at least she should be a carrier like this. She should be a carrier. Carrier means only one, she has one gene effect. The other gene is there, which is why it won't be showing the effect. She will be just a carrier. She won't be having the disease. Yeah. So, at least one should be affected, which is why it should be coming in like this. So, this is also correct. Now, if female has XCX, that is carrier, okay, but males have XCY, then it will be color blind. That is also correct, as you saw it here. Okay, because they don't have another X to overshadow this, right? So, they will be affected. They will be color blind. Men cannot be carriers. Now, the son of a woman who carries the gene has 0% chance of being colorblind. So, that's a son of a woman. For example, this is a woman that we have. Let's make a son. It says 0% chance of the son to be colorblind. Let's see. So, this and this goes, you get X and XC, colorblind female. Sorry, carrier female. Okay. This and this goes, you get XY, normal male. This and this goes, you get XC, colorblind female. This and this goes, this is the important one. XCY, colorblind male. Do you think there is 0% chance? No, Baba, there is 50% of a chance for boy. Okay, if you talk about all the siblings, in that there is 25% chance of a boy to be colorblind, of a child to be colorblind over here. That is a male child. If you're talking just about boys, my dear children, there is 50%. There are two boys, or that one of them is colorblind, so 50% of the times this would be the true statement. Okay. How many colorblind children are there? This child is colorblind and this child is also colorblind. So, I can say 50% of the whole children are colorblind. If I am talking about males specifically, then 50% of the males are going to be colorblind. Works? Now, the son of a woman, okay, so this is the thing that we are having. Yeah, so this would be the incorrect statement. Now, going ahead to the next question. This is a difficult question, that is why it is in red. Cystic fibrosis is an autosomal recessive disease. In an island having a population of 200 people, 98 people suffer from cystic fibrosis. How many people are carriers of the disease? First of all, you have to understand, we just studied cystic fibrosis is what type of disorder, my dear children? It is autosomal recessive disorder. Autosomal recessive disorder, right? So that means both the parents have to give the genes. So both the parents have given the genes, which is why 98 people suffer from cystic fibrosis. So I know that 98% population, my dear children, they have the recessive genes. They have what? The recessive genes. So if you know Hardy-Weinberg principle, it says that you have p square plus 2pq plus q square. Q square represent those organisms who have both the recessive genes. So here q square is going to represent this 98. But how do I exactly find out the frequency of it? How do I exactly find out the frequency of the allele? For that I have to do something. So 98% or 98 people out of what? 200. Out of 200, my dear children, they are affected. They are affected. So if I divide it, I'll be finding 0.49 percentage. Okay, 0.49 percentage. Now, if I want to know the q value, so the q square is what? 0.49. So q value will be what? Square root of 0.49, q will be 0.7. Q will be 0.7. If q is 0.7, what is p? What is p, my dear children? Oh my god, that is my hair. Who have touched things? Just a second. Give me a second. Yes. So how do I find p over here? Basically, I just have to do this. P plus q is equals to 1. So basically, what do I do? If q is 0.7, 0.7, then p is 1 minus 0.7. That will be p is equals to 0.3. Okay, that is 0.3. Now, what do I have to find? I have to find the people who are carriers. So carriers means p, q, both should be there. Both the alleles are there, right? So how do we find that? Always remember, capital T is represented by this. Small t organisms are represented by this. Capital T small t number of organisms are represented by this. When you have both the genes, it is represented by 2pq. So 2 into, what is the value of p? It is 0.3. What is the value of q? It is 0.7. What is it? 0.21 into 2. 0.42. Yeah, it is 0.42, my dear children. This is what? 2pq's value. This is 2pq's value. And how many people, this is the frequency of that. This is the frequency of that. And now if I have to find how many number of them are there, I have to do into with what? 200. When I multiply it with 200, what will I get? I'll get 0.84. No, it will be 84, not 0.84. Into 200 I am doing, right? So it will be what? 84. My dear children, we started off from here. We knew the number. Okay, 98. We had to find the frequency, which is why we did what? Out of the total, we divided it. You got 0.49. That is the frequency of what? Q square. You want to find the value of Q? You do the square root. You get 0.7. You found the value of P then. Okay, then you put it over here in 2pq. Now, you got the frequency. You got the frequency of 2pq, the organism that has having both the genes. But if you want to find the number, you have to multiply it into 200. You get 84. So 84 number of people are the carriers for this particular disease. Tell me, isn't it interesting? Isn't this particular question very interesting? I found it really very, very interesting. Okay, which is why I put it over here. I mean, I didn't. The people who made it put it over here. So I was kind of happy with that. Yes. Now question number 32. Identify the correct sequence of stages in the evolution of modern humans. Neanderthal man, Cro-Magnon man, Australopithecines, Homo habilis, Homo erectus and modern human. Oh my god. Okay, so I always say you have to remember this way. Okay, we have two type of organisms in the initial 15 million years ago. Ramapithecus and Dryopithecus. Dryopithecus were more ape-like. Ramapithecus, Ram, name as the name suggests. They were more human-like. Okay. Now from Ramapithecus you got Australopithecines. Yeah. From Australopithecines, you got Homo habilis. The first hominid-like features were found in the Habibis. Homo habilis. Okay, after Australopithecines. So over here, I do not have Ramapithecus or Dryapithecus. So I'll say Australopithecus should be the first one. So third one should be the first one. Okay. Now from Australopithecines, I got Homo habilis, the Habibis. Both of them, my dear children, they didn't touch meat. They were they were vegetarians. They ate fruits. Okay. Now from them came Homo erectus. Homo erectus, they started eating meat. So it was 5. And then from Homo erectus come Neanderthal man. Neanderthal man. Okay. Now from Neanderthal man came Cro-Magnon man. And from Cro-Magnon you got Homo sapiens, that is modern humans out there. So I'll say, so what is your answer? 3 and 6. 3 and 6, all of them are ending with 6. Okay. 3, 4 and 5. 3, 4 and 5, I just have these options, I'll cut them down. 3, 4 and 5. Now 3, 4, 5, 1, 2 and 4. 1, 2 and 4. So this should be your answer. Okay. This is 32nd question. Now, going ahead with the next question. Select the incorrect statement among the following. Members of phylum Ctenophora are known as comb jellies. Are they known as comb jellies? Yes, because they have 8 bands like this. Yeah, and it looks like comb. The cilia that they have over here, it looks like comb, which is why it is known as comb jellies. Correct. In all chordates, heart is ventral towards the front side. Is it like that? In all chordates like us, do we have heart in the ventral position? Yes. And non-chordates, they have it in dorsal side if they have it. Earthworm and Nereis are monoecious. Wrong. Monoecious means, wait. Wait. Earthworms and Nereis are monoecious. That means both the sexes are in one organism. Earthworm has Nereis. Okay, Nereis is dioecious. It is what? Dioecious. It is not monoecious. Earthworm is monoecious. It is hermaphrodite. Both the sexes are in one organism. But Nereis is not. Always remember that. This is different organism. Yeah. Now, body of Balanoglossus is composed of proboscis, collar and trunk region. Yeah, because they are coming under hemichordata. They come under hemichordata, which is why they have a body like that. But this statement would be wrong. C is totally wrong. That is, earthworm and Nereis are monoecious. Works? Now, special venous connection between liver and intestine as well as kidney and the lower parts of the body are present in frog, and it is known as hepatic portal system and renal portal system, my dear children. So always remember, you have liver, you have intestine. So usually what happens, you have artery opens, arterioles open, you have capillaries open, you have venules open, you have veins at the end. This is vein, this is artery, in between you have capillaries. So what happens over here, in the case of hepatic portal system, from the intestine, the veins carrying the things would be coming. The deoxygenated blood, it will be coming. But along with deoxygenated blood, in intestine, maximum absorption of things also happen. So all the important things, for example, glucose and all, got absorbed. Now this guy, the vein, it reaches the liver, and it opens into capillaries, but later again joins back to form the vein itself. It's not forming the artery, it will form the artery later on. It is forming the vein itself. Over here, maximum exchange of gases, sorry, not gases, the glucose and everything happens, and apart from that, all the important nutrients happen. Okay, and then again this vein will be going to some other organ where there will be opening. If they want to, they will be opening, otherwise they will directly going into what? The heart. So this is known as hepatic portal system. Hepatic as in you are going to the liver, you are transferring something, that is what is known as portal system. So this is hepatic portal system, and you also have renal portal system that is there between the kidney and the lower parts as well. Okay. Next, which of the following option is incorrect? All the elements present in the sample of Earth's crust are also present in the sample of living tissue? Almost, yeah. The relative abundance of carbon and hydrogen with respect to their elements is higher in any living organism than in Earth's crust? Correct. The percentage weight of nitrogen in Earth's crust is very high in comparison to human body. My dear children, atmosphere, I do definitely agree, nitrogen is way too much, it's 78% and things like that. But in Earth's crust, the amount of nitrogen is comparatively lesser if you're comparing with human body, because human body has DNA, it has RNA, it has proteins, all of them have nitrogen in it, so it is having more. Human body has more than what the Earth's crust have. Okay, so always remember, the percentage of nitrogen in Earth's crust is very low in comparison to human body, not high. This would be wrong. If you're asked about atmospheric nitrogen, that is way too much. Okay, that is obviously way too much. Now, the percentage weight of silicon in Earth's crust is very high in comparison to human body. Obviously, we do not have silicon inside us. So, yeah, that's correct. Next, angina occurs due to angina or angina, my dear children. Or occurs, angina is what? Chest pain. Okay, why do we get chest pain? Why do you get chest pain? Because the muscles, the muscles, the cardiac muscles which are there, they are not getting enough amount of oxygen. And when they do not get enough amount of oxygen, my dear children, they become fatigued, which is why you get the pain. So, benign growth in heart, no. Decrease supply of oxygen to the heart muscles. Yes, that is correct. Deposition of carbohydrates in the artery. First of all, it's not carbohydrates, it is cholesterol, lipids that is getting deposited. Okay, but that is not going to cause angina. It will cause, but not directly. It is this one that causes angina. Now, increased blood pressure. Increased blood pressure obviously would be damaging your heart. Okay, after some point of time, but the actual reason will be this. Now, during expiration, diaphragm becomes what? During expiration, as in when we are breathing things out, air out, what happens to diaphragm? My dear children, when we are breathing in, we need more space for the air to be there, right? So, the diaphragm which was like this, dome shaped, it becomes like this, it becomes flattened. It becomes flattened. But on the other hand, when you are expiring, when you are moving out the air, at this point of time, it is going to move up. From here, it is going to move up. It becomes dome shaped. It becomes what? Dome shaped. It is not flattened, dome shaped. Now, in the measurement of blood pressure, the denominator and numerator represents. You know what? I messed up with this question first, when the first time I solved it. It's a so easy question. Still I messed up. I know. You might also mess it up, which is why I am telling you to read the question very carefully. In the measurement of blood pressure, the denominator and numerator represents. So, at the top you have systolic pressure. Bottom you have what? Diastolic pressure. Systole means contraction. Pressure on the walls. Okay, pressure on the walls of the blood vessels when the heart is contracting. When the heart is contracting or pumping, my dear children, the pressure that it exerts on the wall, that is the systolic pressure. Diastolic as in when it is relaxing. So, over here, denominator is this and numerator is this. You are asked about the blood pressure on the denominator and the numerator represents. So, first you have to talk about this and then you have to talk about this. So, it cannot be systolic, it cannot be pumping and resting because systolic means pumping, diastolic means resting, so it cannot be that. This is A, this is A. Resting and pumping pressure, denominator and numerator. So, I know I went with this first, then I realized something is wrong. So, then I went with this. So, it's because I didn't read the question carefully and I do not want you to have the same trouble. So, please my dear children, please read the questions properly. Now, question number 39. Which of the following is abundant in red muscles? The red muscles, what is abundant, my dear children? Red muscles are found in where? Heart, right? So, over there, what do you find? What is more? Myoglobin and mitochondria. So, myoglobin, basically what it does is not like hemoglobin, okay, but myoglobin has very high affinity to oxygen. So, basically the hemoglobin that carries oxygen, myoglobin stores this oxygen. It is like a reservoir of the oxygen. Whenever the muscle cells need it, it gives. Okay, and it has enough amount of mitochondria, my dear children, because mitochondria, you know, with the help of that only contraction, relaxation, everything is be happening. So, which of the following is abundant in the red muscles? It is myoglobin as well as mitochondria. Works. Next, which of the following hormones never reach to the cytoplasm? So, this is a cell. This hormone never reaches the cytoplasm. What is that? Estrogen, FSH, progesterone, testosterone. First of all, I will tell you this and this. Three of them are steroidal hormones. Steroidal hormones means they can easily pass through the cell membrane because a cell membrane is also made up of lipids, right? So, it can easily pass through. But FSH, it's not a steroidal hormone. It is what? A peptide hormone. A peptide hormone. And peptide hormone, do you think it can pass through the lipid? No. Basically, they will be having receptors outside, they will come attach to it and then activate the intercellular things and things like that, but they themselves are not going to get inside. Okay, so always remember, FSH is a peptide hormone, similar thing goes with insulin, similar thing goes with glucagon and things like that. So estrogen, progesterone and testosterone, they are the steroidal hormones that we have. Cool. Next, 41st question. A woman had an extreme premature delivery. Okay, she gave birth to a baby boy at 23 weeks. 23 weeks. 23 weeks means how many months? 23 week divided by 4. Every month has 4 weeks, right? So it is almost 5 point something something. So or basically around 6. Around 6. It has not completed 6. Okay, 24 weeks is basically say you completed 6, but 1 week less. So 5 point something something. Okay, now the birth weight of the baby was 950 gram. Which of the following is most likely to be observed in this baby? Now if 6 months are not properly being done, what happens at the end of 6 months according to NCERT? After the end of 6 months, eyelashes form, eyelids separate, and then there will be hair on the body. So these 3 features, okay, any of these 3 features could be missing. So baby's heart will not be adequately formed. Arey baba, this is formed. Okay, the baby's limbs and digits, this happens after 1st month, this happens after 2nd month. Now baby's external genitalia will not be adequately formed. This also happens after 2nd month. The baby's eyelashes will not be adequately formed. This is the one. So for every month you should be knowing this is how they ask the question. They twist the question and they ask you. So please make sure you're knowing this. Yeah? Yes. Next, one of the following is not true of hemophilia, for hemophilia. What is it? Royal disease. Is it a royal disease? Why do you call it royal disease? Because Queen Elizabeth was having this disease and she passed on to the other, lineage she had. So it is a royal disease, correct? It is bleeder's disease, yes, because the person who has hemophilia, they would be continuously bleeding if they have one wound, because the clotting factors are being damaged, the genes coding for clotting factors, they are being damaged, which is why it is known as bleeder's disease also. It is X-linked disorder. Do you think it is X-linked disorder, my dear children? Yes, hemophilia is an X-linked disorder. It happens, the gene which is responsible for the clotting factors, it is on the X chromosome, so it is affected. Yeah, so that is true. Y-linked disorder, no, it is not Y-linked disorder. This would be the wrong statement. Rest all of it is correct. Now, when more than one adaptive radiation appear to have occurred in an isolated geographical area, it is what? So more than one adaptive radiation. For example, over here, this geographical area, I had Australian marsupials. Okay. Now from them, some organism went that side, some organism went this side, some organism went this side, some organism went that side. All of them would be having different attitude because they are staying in different regions now. Agreed? So this is divergent evolution. They are diverging and they are evolutionary. They had a common ancestor, but they have evolutionized differently. They will have different features. Yeah, structurally they might be same, but they would be having some differences in them functionally. Same place. Placental mammals also came in. Australian marsupials were there, but now placental mammals are also there. They are also, one guy moves there, one guy moves here, one guy moves here, one guy moves there. Now basically, the 2 guys which moved there belonging to 2 separate communities, my dear children, they would be having same features. Why? Because they are staying in the same habitat. Okay, all the ones that flies would be having wings, right? Similarly, all of them would be having similar features over there. This also similar feature, this also similar feature, this also similar feature. So can I say they are converging together? This is convergent evolution. They are converging to a common point, my dear children, which is why this is an example of convergent evolution. So when more than one adaptive radiation, when more than one radiation happens, okay, adaptive radiation happens, it is known as convergent evolution. If only one is happening, it is known as divergent evolution. So this becomes your answer. Cool? Okay. Are you? Yes. It is not divergent evolution because you are not talking about one. Again, it's not natural selection, it's not genetic drift. Genetic drift is basically if we are changing the population frequency and it is occurring by chance. Okay, for example, I had so many green beetles and the white beetles. I accidentally stamped on the green beetles, all of them died. I had the white ones. So this is, the frequency is changed because of what? Because of chance, and that is the example of genetic drift. Now, a disease caused by autosomal primary non-disjunction is. Non-disjunction means basically they are not able to move apart from each other. They came together. So autosomal means it is happening in the autosomal chromosome. Sickle cell anemia, my dear children, it is a sex-linked it is an autosomal disorder, it is a recessive disorder, but they have nothing to do with disjunction. Basically, disjunction is when you have number of chromosomes, they becomes increased because they are not moving apart from each other. So sickle cell anemia happens because there is a chromosome, there is one gene that is not properly being produced, which is why this happens. So that has nothing to do with non-disjunction. Down syndrome, yes, why I'll tell you. So you will get one chromosome from one mother, your mother, okay, 21st chromosome. Same, you will get another chromosome from your father, 21st chromosome. But over here, in the case of Down syndrome, what is happening is that either one of the parents are giving you 2 chromosomes because they didn't move apart. So usually from a pair of chromosomes, you get one of them. But here both of them are coming together because of failure of non-disjunction. This what? It didn't move apart, my dear children. And it is happening in the autosomal chromosome, my dear children. 21st chromosome is autosomal chromosome. We have 23 pairs of chromosome. The 23rd pair is basically what? The sex chromosome. The sex chromosome. 22 of them, they are what? Autosomes. Okay, so 21st until 21st. So if this is 21st chromosome where you're giving an extra copy, this would be Down syndrome. Okay, now Klinefelter syndrome is an extra additional of X chromosome, but that's a sex chromosome, my dear children. You're talking about autosomal, so this won't be the case. Turner syndrome, where basically you should be getting one X chromosome, but you're not getting it. So there is loss. Okay, so that is also a case of non-disjunction, but it is not autosomal. Both of them are sex linked. So only answer is your Down syndrome. Okay, I have some pain over here, which is why I'm not able to speak much. Yeah, 45th question. There is a forest next to Raju's village, okay, which is a natural habitat for all the endangered species of lotus. Okay, the government plans to develop a highway through the forest, which will cause disturbance in the habitat and may lead to extinction of the endangered species. That is a problem. Being an environmental activist, what must have Raju suggested the government in order to save the species, as the highway project is also important for development of the village. So highway is important, but you have to save the species also. So what is the conservation method that probably he might have suggested? In situ conservation, migration, ex situ conservation, deforestation. Not deforestation, not migration also, because you want that place to develop. It's not like everyone should be going out, right? Ex situ conservation, basically where you're taking the organism and putting it elsewhere, but when you put that way, not all of them will be safe, and you cannot put all of them, right? So what is the probable thing that you can do is in situ conservation. Is what? In situ, wait. May lead to extinction of endangered. Okay, wait. Question. After marking, I get it wrong. Okay, listen. Over here it is said that it may lead to extinction of endangered species. So that means they are endangered. If we leave them in their own area, there are high probability that someone could come and eat them, or some organism or some bus or train or anything could go kill them. So we do not want that to happen. They are endangered, and when someone is endangered, what do? You're going to take them out from their habitat and going to put them in some other habitat. Okay, create something like that and going to conserve it there. We are not going to conserve in their original habitat. We are going to conserve them elsewhere, and that is why it is known as ex situ conservation, not in situ. So this becomes your answer. I'm so sorry for telling this is the answer, but yeah, since here endangered species and everything is being mentioned, you cannot put them in their natural habitat. In the natural habitat, they could be getting killed by other organisms or by human beings or by any other activities. Since they are endangered, we have to put them in other areas where they can be given more care, personal care, and that is ex situ conservation, which is why this becomes your answer. I hope it makes sense. Children, you don't have to mark this up because I just taught you, but please try to understand what I'm trying to say. So since this is there, your answer have changed. Unexpected change, but yeah. Yes, and with that, we go ahead. That's all. So I hope, my dear children, we are able to help you. Do let us know if we are able to help you. We will be really, really happy, especially me. I'll be really happy to help you down. If you feel that I have not helped enough, okay, please put it down in the comment section. I'll be solving your doubts as well. Okay, please reach out to me just like a normal friend of yours, yeah, and I'll be here to help you out. Children, do not forget to subscribe to the channel, also like this video, and also comment down, and also share it with your friends, and please register for the free mock test if you have not applied it yet, okay, because we are not going to provide you solution videos for all of them. So that is not the way how you would be seeing this question papers, okay. You have to do it by yourself, because almost okay, so we thought that we will be providing you video solutions for 2, and we have almost done it. If we need a third one, we probably do it. That depends, but you should be attempting the question. If you want to refer to the answer, it will be also given there where you download the question papers. So make sure you are attempting it and checking your answers as well. Works. So that's all. Wishing you all the best. Bye bye. Take care.